Hey guys, so today we are going to talk about the gram-positive bacteria. Now, if you don't know we divide bacteria into two categories, into gram-positive and negative, on the basis of a stain that was developed by Christian Gram. We'll talk about how this stain exactly works in a while. So first of all, let's talk about bacteria. So if you know, you already know that all cells, because bacteria they are also cell, so they will have a cell membrane underneath, right? But just above the cell membrane, they have the cell wall. Now, if you look here, this is the cell membrane, and as you can see, this is the lipid bilayer that we always talk about. In gram-positive bacteria, they have a really thick cell wall made up of a chemical called peptidoglycan, also called as murine, right? Now, the basic difference between gram-positive and negative is the length, the width of these layers, and in addition to a very thin a very thin peptidoglycan layer in gram-negative bacteria, we also have another membrane called an outer membrane. We'll talk about this in a little bit more detail when we talk about the gram-negative bacteria in another video, but right now we want to talk about the gram-positive bacteria. Let's first talk about the peptidoglycan structure. So biology, it's very simple. You just read something backward. Glycan, carbohydrate. Peptido, a chain of amino acids, right? So yes, there's a carbohydrate derivative and there's amino acids in the structure. So what are the carbohydrates? We have N-acetylglucosamine called NAG and N-acetyl muramic acid called NAM. These are found in alternate fashion and arranged helically, right? So you can assume this yellow string to represent the NAM and NAG chain. So this is, if I want to just represent, this is NAM and NAG chain, and I can write NAM, NAG, NAM, and so on, right? Now, so this forms a skeleton of the peptidoglycan molecule. Now, you'd be wondering what are these streaks, right? These streaks coming out. These are peptides, right? Peptides is a group of amino acids, chain of amino acids. The second component of the peptidoglycan structure is amino acid. So usually you can find 4 amino acid short chains. We have 4 amino acids forming the peptide short chains, and those are the alanine, the glutamine. Now, in most of the gram-positive bacteria, we have mesodiaminopimelic acid. Very important to understand. Some have lysine with them. Now, what we also need to understand here is DAP. DAP is actually found in a lot of gram-negative bacteria, but a few nocardia. So how do I remember this? Noca lives in bacillus, back. Noca lives in back, around the corner. Corynebacterium. So these bacteria generally they stain positive, but they still are somewhat like the negative. They have this DAP with them, right? Now, what about the linkages? So we can see that there's a NAM and NAG chain, right? That is how the helix is formed. And there is a 5 glycine residue. Now, GLY stands for glycine. So this is um, the alanine, this is the glutamine, this is the lysine, and this is the alanine, right? Again, now we have 5 glycine peptide linkages between the short chains that link in the gram positive bacteria. So you can see that this is a type of linkages found in gram positive. It will change in gram negative, right? So sometimes you have benzodiaminopalmilic acid, sometimes you have lysine, and in some gram positive bacteria you also can have DAP as the short chain. So this is about the core structure of the cell wall in the gram positive bacteria. Very important, notice the presence of D amino acids, because our enzymes are trained to digest the L amino acids. This the presence of D amino acids kind of helps the bacteria to protect themselves against the enzymes. Now, in addition to that, we have some more stuff that we should know about. So this is our whole plasma membrane, and that is the cell wall. Now, one thing that another compound that actually helps them is teichoic acid, which is a polymer of glycerol, which is joined by phosphate. So you can represent them as like that. So glycerol phosphate, or ribitol joined by phosphate groups. Teichoic acid can be linked to the plasma membrane, as you can see, linked to the plasma membrane or linked to the cell wall, right? They can be linked to cell wall as well. Kind of it gives them a negative charge. Now it can be involved in binding to the host tissues. Sometimes they are also thought to protect from the harmful substances. Now comes a question, why do they stain positive? So for the gram staining, first we stain the specimen with the crystal violet stain, also talked about as the gram stain. Once the gram stain is there, we add iodine. Iodine is a mordant that forms a complex which allows this to stick in the cell wall and the membranes. Further, we add alcohol. Now, alcohol is assumed to close the pores of the peptidoglycan wall. This helps in preserving the stain into the cell wall and membrane. So, when we try to decolorize, the decolorization does not happen well. Why? Because they are very tightly locked in, and finally they will look violet. They will look violet like that. Gram negative bacteria have very thin cell wall, and hence they cannot interlock this. Further, because of an outer membrane which we discussed in the beginning, the alcohol kind of makes enough pores in the membrane which allows this iodine and crystal violet complex to leak through. That is what allows it to be decolorized very easily. And then when you stain them with a counter stain, which in this case is safranine, they stain pink, right? So that is why the gram positive bacteria, they stain positive. I hope this makes sense.
We hope this video was helpful. To see more biology topics covered, click on our channel and don't forget to subscribe.